Thank you for allowing me the opportunity to speak today. And we're really, I think the discussion of detective work is really very much on topic for what I will be describing today, because we are in the business of trying to understand how different pathogens might be implicated in brain disorders. And this is sort of an irreligious concept for quite some time, but even in the 1830s, people were beginning to have the idea that perhaps there may be environmental factors, including infection and other types of things, independently of moral or even immoral causes, that there may be changes in brain disorders. And that concept has come up, but then been submerged again, and then come up again. It's almost as if we've had a decapitated body, where the head has had its own special province, that has been independent of the laws of biology and physics. We look at brain disorders as perhaps best understood as the intersection of three separate factors: genes, environment, and timing. And these factors interact both before the development of the fetus and also after the development of the fetus. And depending on when these interactions occur, the timing of a particular exposure and its interaction with the genetic factors that are unfolding either during development or through later years in development, we can have outcomes ranging from childhood disorders like autism, attention deficit disorder, and early adulthood having ME/CFS, mood disorders, and so forth. And even in late life, Parkinson and Alzheimer. And sometimes these are disorders that are laid down by what happens to the fetus during the mother's pregnancy. So in utero. And these environmental exposures may not only be limited to infection, but could also be xenobiotics or toxins, nutritional ones, as well as even psychosocial stressors. These all have physiologic, biochemical effects. The example is made very clear to me through this kind of fun example. In a sense, I feel very relieved that it's only adolescent onset cannabis, not earlier, not later, that in conjunction with a gene may lead to a psychotic disorder. And you can see here, these are the significant values: not significant just to have the gene problem, not significant just to have the early onset cannabis use without the genotype, but the combination leads to a very significant increase in the brain disorder. This is a concept that comes up time and again in how we model disorders involving the brain. When we look for causes, potential causes for brain disorders, and you look through the literature, there are innumerable factors, innumerable viruses, bacteria, and even seasonal factors that may be associated with infection, such as being born in the winter, or dysnutrition factors that may lead to immunosuppression and increase the risk of infection. There are so many of them, it's highly unlikely in our mind that only a single agent would be responsible for the very many cases and the heterogeneity that you see in ME/CFS. So how do we go about this? It's like we have crime scene investigation, the CSI story. How do we go about looking at this? Some of it can be modeled in animals using bacterial challenges that are like lipopolysaccharide, double-stranded RNA, a viral mimic. And we can look to see manipulating the genes, the timing, and then looking carefully at the physical as well as the more cognitive types of outcomes. Of course, in an animal model, that's always a challenge. We know that many disorders, not just ME/CFS, but a whole host of disorders are now thought to be related to infectious factors and or immune factors. And so, again, it's important in my mind to remember that infection is just one way to disturb an immune system. It can happen through psychosocial stress. It can happen through trauma. It can happen through a toxin or a combination thereof. And even in genetic disorders that are thought to be, where we think of these affecting the brain, Rett syndrome and Down syndrome, these also have immune disturbances that are associated with them. So it can arise from the genetic side alone as well. You saw a little bit of this with Dr. Towers in terms of how we go about in this in this area where we have highly sensitive methods. How do we go about trying to figure out what relationship there might be between a particular microbe or infection and disease? And there were Koch's postulates that said basically one-to-one association. You had to be able to reproduce it in animals. But we know, first of all, that the host, the individual in whom the disorder, in whom the infection has occurred, has an enormous amount of control in terms of what the outcome looks like. And then again, we need to look for various molecular markers. However, those are overly sensitive in some ways. And with some of the techniques that we use, which are amplification techniques, the polymerase chain reaction, PCR, this will also amplify noise and not just signal. So we have to be very cautious therein. We also have the similar dilemmas in autoimmune disease, and many of us are beginning to develop some body of evidence that suggests that perhaps ME/CFS, or at least a subset thereof, may be related to autoimmune disorders. And it's a very similar type of phenomenon as with Koch, as with Tepsky's criteria, but this was revolutionary in 1957, and in fact, the concept of autoimmune disease having anything to do with a real disorder, the idea that you would actually have a reaction against yourself, that your own immune system would attack your own tissues, was really just unheard of. In fact, people laughed at it and called it the horror autotoxicus, and just really poo-pooed the idea. We know that now that this is very important phenomenon, but we do know that there are many other things that we need to look at. It's not just the one-to-one relationship that we need to challenge of an infectious agent or an autoantibody to disease, but a larger picture that we may not always have the early evidence that we need in order to complete our detective work. It's like the CSI agent coming in and 6 years later, and there's nothing left to really, or very, very scarce traces of what had happened. So we have to be able to be prepared for looking at long-term sequelae. Perspective studies can be very helpful in this regard, but also trying to understand what types of molecular footprints, what types of processes would be able to tell us, if we imagine how we got to the end point, what are the processes that we know that could get us there. So I just want to very rapidly go through a number of various ways, just to give you the point that there are many ways in which an infection could potentially lead to disease. So these are ways that we need to identify, to look in animal models, to look in human systems with samples that we have, to allow for the possibility that perhaps it's not just your straightforward infection. Not everybody who has ME/CFS, even if it were due to a particular virus, not everybody's going to come to their physician and say, I'm sick, and then, let's prove that I have an infection now because 6 months from now I'm going to have a chronic course. We just don't have that. We need to be thinking about ways in which you can have a toxin, you can have things that do happen quite locally and directly, but you can also have host responses where the immune system is involved in the disease outcome. You have immunosuppression that perhaps is a setup for additional infections that may lead to cytokine changes, and which in themselves can cause cognitive and fatigue, sickness behavior type of reactions. And we also know that sometimes it's only in certain critical periods of life that these factors may lead to the effect. I guess a good example there is rubella virus having an outcome of birth defects with a first trimester exposure, but not in other time points. And we are now becoming more and more aware that there are a variety of ways in which we could have an infection affecting functions like hypothyroidism. Some people have found evidence, for example, that thyroid function may be affected by HHV6 infection. And so there are a whole host of ideas that we need to be able to pursue, or at least keep our minds open to these possibilities. We also know that sometimes molecular mimicry, that the agent itself, either a bacterium or a virus or a parasite, may mimic, have proteins, stretches that resemble parts of the body, resemble parts of the brain, sufficiently so that they can create an antibody that binds up that part of the body and alters its function. It can alter your energy level, your ability to pay attention to things through effects in the reticular activating system, in various brainstem nuclei, and so forth. And also we need, of course, to be able to have our eyes open to the idea that it could be a hit and run. Long-term effects of infection. How do you do, in multiple sclerosis? If this was an exposure that was in early years, perhaps the agent is gone, but you can look at certain epidemiologic factors. The latitude where you lived before puberty is a factor that increases your risk. If you live in northern latitudes, it increases your risk. But if you live in southern latitudes before puberty and then move to northern latitudes after puberty, you don't have that same risk. So the same geographic region and whatever exposures happen there, it's only during a certain time window, and you need to be able to think about those types of things and ask the right questions, or at least allow for the possibility of the different questions. We also now know that not every infection is bad for us. In fact, we need our bugs. We have more of them in us than we have our own human cells, and we need our commensals, we need our normal gut microbiota in order to allow for normal brain development and behavior. And this is a very important factor. Normal sleep is dependent upon having a normal bacterial community in your GI tract. You don't sleep properly unless you have peristalsis, allowing some of the products of those bacteria, shooting the little bits of endotoxin, bacterial products into your bloodstream, increasing your cytokines, and putting you into slow-wave sleep, your restorative, healthy sleep. We know this from animal models. We also know that the intrauterine environment, what happens to your mother during pregnancy, may affect your risk for even things from depression and perhaps even chronic fatigue syndrome. Not necessarily the only hit, maybe multiple hits. And here we are showing the relationship between the mother's peripheral blood cytokines, IL-1 beta, IL-6. We see that in the women who have depression, in the green line here, we have a hypoperfusion in fMRI in the stress response brain circuitry. So as the cytokine inflammation levels go up, you see this decrease consistent with what we normally see in individuals with depression. It's sex dependent, tends to be more in women. There is a sex difference, of course, in chronic fatigue syndrome as well. And this is the hypoactivity that one sees, just to give you the example. I also want to give you an example of how an autoantibody can affect brain and behavior. This is not necessarily the same spectrum that one would expect in chronic fatigue syndrome, but it certainly includes some of the types of cognitive memory dysfunction that one has observed in chronic fatigue syndrome. This is a disorder that occurs after a simple strep throat infection. It's related to this molecular mimicry process, and it includes obsessive compulsive symptoms, anxiety, as well as tic disorders. And there's some B-cell markers that are associated with this, which are the same markers that show risk for rheumatic fever risk after a strep throat infection. Okay, so you see this little mouse. He's doing backflips. What we've done is we've given him the strep, a strep vaccination, if you will. It's dead strep, and the animal goes into backflips and does these backflips obsessively over many hours. What you see also is the development of brain antibodies, IgG, that are in the mouse, in the brain of the mouse. And you can see these in vitro as well. When you take the peripheral blood, these stain against neurons. It's primarily in the basal ganglia, but also in the cerebellum and other regions that are known to be affected in chronic fatigue syndrome as well as in autism models as well. This is what happens in the control. In the red here and blown up, you see that these are the deposits that occur when we take the blood of the animal that was directly injected and immunized with strep and had an autoimmune response to it. We took those antibodies and put it into a naive mouse. And here is the binding in the brain that one sees. Then there are behaviors that also occur in conjunction with that. This is a social investigation, is a test where it's called the resident intruder task. The animal who is the resident of the cage should be territorial, marking the territory and, sort of resisting any invaders. But in this mouse model, the antibodies, which is the strep serum that has been injected, you see this decrease in social interaction, and you also see more defense escape behaviors as well. And if with the control, you don't see this. And then if you take the IgG out, depleting it before the injection, you see that you have returned to the normal levels of social interaction and the normal defense escape parameters. And we see similar phenomena with cognitive tasks that we have performed. What is the challenge is figuring out exactly what the host protein is that antibody induced by the strep is responsive to. And so we found through various, we had complex techniques, non-denaturing immunoprecipitations, we were able to identify that a variety of immune molecules and oxidative stress heat shock proteins were involved, heat shock protein 70 in particular. And this is just to show you the potential of what we might be able to do in a scenario where we had a model similarly, perhaps, for ME/CFS. We took the target that we found in the mouse. We cloned the human homolog. We got a commercial antibody to that HSC70, and this is in an in vitro model. We took pandas, which is what this syndrome is called. We took serum from children from our National Institutes of Health, and reacted those. You see a very similar pattern, and there was no reaction in the control. So this is something that could potentially be applied, and we hope, to this scenario. Infection leads to a host of oxidative stress reactions that make these proteins, these heat shock proteins, more available for an autoimmune response to be generated against. But it's not just infection. Infection is just one. There's toxins, heavy metals included, radiation, other sources of oxidative stress, reactive oxidative intermediates. And we know that there are certain ways in which one can actually buffer against that using various antioxidants. We know also that there are cytokine changes that occur along with these. These in and of themselves can have effects. This is called the tryptophan degradation pathway. The source of most of your tryptophan is from your bacteria in your gut. If they're not doing their job, you're not going to have the right amounts of these. You're also going to have degradation down this pathway, which is a normal pathway, but if it is in excess, it can lead to excitotoxicity, loss of brain cells, or dysfunction therein. And it can also reduce the ability of your synapses to function to lay down memories. So we think about these as very important processes that are likely to be involved in a wide range of neuropsychiatric disorders that involve both fatigue as well as cognitive dysfunction. But when we're trying to figure out really what agent might this be, in our laboratory, in our hands, when we have made these pursuits, we are very agnostic. We don't come in with any preconceived notion. In fact, we try as best as we can to create an opportunity for the agent to show itself as being an offender. You already saw this from Dr. Towers, but we call this sort of our pathogen de-discovery, so XMRV and chronic fatigue syndrome. We also applied the same sort of technique for born disease virus in a wide range of neuropsychiatric disorders. This is an agent that had also been implicated in chronic fatigue syndrome. And thirdly, pathogen de-discovery part 3, we also looked for measles virus in the intestinal tracts of children with autism, where in the UK, I'm sure I don't have to tell you about the Wakefield story, but we pursued this in all earnestness. We used the original laboratory used by Andy Wakefield, John O'Leary's laboratory. We had a CDC laboratory, and we had our laboratory samples were prepared, of course, coded and blinded. But we found no association between measles and disease. But what we did find was that there was something going on in the intestinal tracts of these children. They had no machinery. Of course, we've thought about diet with children with autism. They, just like 50% of people with ME/CFS, there are GI disturbances in approximately the same amount of children with autism. They have none of the metabolic machinery to digest disaccharides, and even if they have glucose, none of their glucose transporters are working. It's seriously suppressed. And it's in correlation with the bugs that are in the types of bugs that are in their GI tract. So you see this vast reduction in both the small intestine and the large intestine of the certain bacterial communities. So it's all disrupted. We also found an unusual agent called Sutterella, which was only found in the children with autism. In fact, they also had an antibody response to it, suggesting that it was an infectious agent. So this might be a bad offender. What relationship it has to the disorder, we don't know. But it does allow us to have an understanding of what some treatment avenues may be, even if we don't know how we got there. We believe that the GI system and disruptions therein may be critical to the development of autoimmune disturbances. This is a sort of a graphic of your small intestine, the terminal ileum, where enormous amount of important biochemical actions occur. They're affected by both what is in your diet, and the products of the microflora, whether they're good, bad, or indifferent. And we know that there are neurotransmitter effects of glutamate and reaction with glutathione and antioxidant. And the redox status, or the oxidative stress therein, is conditioning the immune system. So we have a scenario where we can have a change in the microbiome, which may be subtle, but may have important metabolic effects, change in tryptophan and and availability of glutathione. And we also could have celiac or inflammatory bowel disease. These lead to an increase in inflammatory cells known as the Th17 cells, the source of your IL-17, and also a decrease in the more protective T regulatory cells, suggesting a trend towards increased inflammation and increased autoimmunity. If you have this happening, you could have bacterial products that are coming in a leaky gut. They could come into the bloodstream, cause increased fatigue and sleepiness, or lack of energy or motivation even, and also alter your cognitive function through the cytokines that are associated with these differences in the cell types. So what are we doing? How are we applying these to the story of ME/CFS, to the mystery of ME/CFS? We have collected a large number of samples in chronic fatigue syndrome through the Chronic Fatigue Initiative, through the NIH study where we had initially looked at the XMRV story with Jose Montoya's group at Stanford, and also some really unique studies, although Dr. Peterson, who you heard earlier today, has been involved in both of these studies as well. We have some specialized studies where we're looking at cerebral spinal fluid that he's collected over many years, as well as what we call an unusual cases study that we're just beginning. So we feel that we have the right samples to begin to do this detective work, and we pursue this with a sort of a semi-agnostic approach. We start with a sort of what we call a stage strategy. We start here with mass tag PCR, a detection technique that does use, looks at the molecular sequence and tries to recognize it. We start with panels that are looking at the agents that have been implicated before in chronic fatigue syndrome, or even if there isn't any evidence for it, even if it was just a hypothesis that somebody raised, or if it's an agent that has been associated in animal models or in humans with some of the features consistent with chronic fatigue syndrome. We've collected a large number of samples upon which we perform this because we don't know what body compartment we should be expecting the agent to still be residing at the time we're coming in and looking. So we are looking at everything from the peripheral blood molecules, cerebral spinal fluid, also nasopharyngeal, as well as rectal swabs and stool. And so we're looking for agents. We're using the high throughput techniques. We're building techniques that will, if we find something that's unusual, we'll build as sensitive an assay as we can and go back and look again, see if we might have missed it, and try to define what is there. We also look at the GI tract as well as other areas to look at the bacterial populations and also the fungal populations, what's called the mycobiome. We need to be agnostic, I think, because we often can be fooled. You don't know what you're not looking for. And we have this example from the influenza season surveillance in the New York City Department of Health. They thought everything was influenza, but they were distressed because they couldn't always find it. And so they actually found that in the influenza season, so in the winter months, we had this lower level of the influenza virus here in January, February, and in fact, what was at a higher level were enteroviruses or rhinoviruses, which is the common cold virus. We also know that we need to understand what's happening in the host. So we're pursuing techniques using a wide variety of samples again, looking at what the gene expression is, using RNA-seq, looking for microRNAs and other small RNA molecules that may interact with the exposures and alter the response to various infections or other environmental factors. And we're also doing immune profiling through a variety of techniques, looking at antibodies to various infectious agents, and also taking a sort of open view, looking targeted proteomics that will allow us to understand all the proteins that are there. And while this is a very preliminary glimpse, I just want to give you a sense of the types of data that we are generating and and hoping to tease apart. This is an example from a large panel, 51 cytokines, chemokines, other immune molecules, and it's just showing you that in cases versus controls, these are the associations of only those significant ones that were less than this 0.0012 level. And you can see here how tightly associated all of these cytokines are with different drivers in the cases. And it's not really important to see each individual label, but rather to understand that there is a very different way in which the immune system as a whole is functioning or dysfunctioning, as you might state it, compared to the controls. And so in closure, I like to end with this example where we are recognizing that we can't close our eyes to new options. We do need to maintain a sense of agnosticism. Doesn't mean we can't have any belief system that or ways to develop a hypothesis and evidence for it. But we used to think that ulcers were stress. And some ulcers may still be stress, but mostly the majority of them can actually be cured by an antibiotic trial, because we know that there are the Helicobacter pylori is a common agent. And this is illustrated very well by this example from the Einstein quote. He was active as a professor, said, the students complaining, the questions of this year's exams are the same as last year. But true, this year all the answers are different. We have new tools, we have new ideas about what may be involved, and we must keep our minds open to these. And this work involves a huge number of collaborators, Dr. Peterson, who's here today, all the clinicians of the CFI and NIH studies, as well as Ian Lipkin, the director of our laboratory, and funding from many sources. Thanks very much for your attention.
That was excellent. Now, we have time just for one or two little questions. Okay, man?
Hello, I'm interested because my daughter, who has ME, has a high ASO streptolysin titer and has been shown to have antibodies to the basal ganglia. She has no movement problems at all. She just has cognition and sleep problems, and it was to the pyruvate kinase receptor that she has antibodies. Do you have any comments?
Well, we know that the basal ganglia also are involved in so-called what we term as motivation. And what is motivation? It's not necessarily that your will, or desire is there. It's really your ability. We look at motivation, it's in some degrees, by the outcome of that motivation. We infer motivation, and so I believe that we know that the basal ganglia are actually very complicated, and you're not describing something to one of the dopamine receptors, which are also present in the basal ganglia. And the pyruvate kinase is an enzyme that's involved in a whole host of functions regulating the movement output that comes out of the basal ganglia. So I think that we need to understand more about because could this be that there's something inhibiting what one may want to do, but you just can't summon up and corral your energy sources or whatever energy you may have, you can't corral your muscles into moving. We know this is similar in a disorder that used to be thought to be related to influenza, but we think is also related to strep, or there's some good evidence for that more recently, something called Encephalitis Lethargica. Maybe some of you have seen the film Awakenings, for example. These individuals are sort of unable to move. So it's not that they have tics. It's really that they're unable to sort of summon everything to actually execute these things. So I think that it's not necessarily something that is just a comorbidity or a red herring. It may have a relationship to one part of the spectrum of what we see in ME/CFS.
Thank you.
Well, are the antibodies treatable?
There are many thoughts on that. So the example that I showed with the heat shock protein 70 in the in vitro, where we were looking at the pandas children, those children were going for plasmapheresis. That is one treatment that some people have tried. I'm not recommending a particular treatment right now. Intravenous immunoglobulin is another. Some people have tried steroids. Some people have used antibiotics as prophylaxis for additional episodes so that you don't keep rekindling the process. But it is complicated. Do I think that it is treatable? I do think that there are ways to treat it, but we have a long way to go, and we need to understand these much better. Some evidence that some antibodies may require certain oxidative stress in order to actually remain bound. Could strong antioxidants play a role? Maybe. Again, all speculation really deserves more careful research.
I'm intrigued by your Sutterella. Could you say a little bit more about that? Is it really unique to autism? And if so, what are the consequences of treating autism?
Yeah, it's a very, very important. We don't know what it means. It has only rarely been reported. It was first described at the research, the main New York State Research Center. So it's Sutterella Wadsworth. This is the Wadsworth Center. There are a few strains that are known. Only been described in pathogenic situations like inflammatory bowel disease, so we don't think that it's something that would be unique to autism. And this is the subset that we looked at there. These were children who had autism with severe enough GI disturbance to warrant, on a clinical basis, colonoscopy. So we didn't bring them in on a research basis for colonoscopy. We made use of children of samples from children that were already going for colonoscopy because their GI disease was that severe. So at best, I think all we could say is that this is something that is really worth pursuing in children with autism who have severe GI disease, and then to see if it confirms. It's also intestinal biopsy material. We don't see it in the stool. In other populations, so in the other reports have all been on not on intestinal adherent microbiota, but rather on the microbes in the stool, which is a different story. Those are the ones that aren't hanging out and perhaps not affecting metabolism to the same degree. We don't know, and we are pursuing this, and it's a very active area of investigation in our laboratory.
What about possible analogous treatment to the gastric ulcer that Martin developed with antibiotics? Is there any prospect of that?
I think that would be really a great thing to do. We're always very cautious because we already know that the microbe communities in the gut appear to be disrupted, and the last thing we want to do is to create a new disturbance, and it could be secondary. We don't know whether it's primary or secondary. I think first we really do need to understand the extent, and we developed using some relatively new techniques. We're in the midst of developing and validating a blood test that will tell us whether antibodies to Sutterella are present. The children who were in this study, 100% of those who had the Sutterella agent present on molecular tests had antibodies to Sutterella, suggesting that it really was an infection and was recognized by the host immune system as being a foreign invader. Some who did not have evidence in the molecular tests did have antibodies in addition, but 0 antibodies in the controls. 0 of the controls had any molecular evidence of Sutterella. So I do think that it is associated at least with the GI disorder. Is the GI disorder an autism comorbidity, or is it a subtype and a phenotype, a subset of the disorder itself, a different type? And that's the same issue, in chronic fatigue syndrome. It's such a heterogeneous disorder. Is the GI affected subset just having comorbidity, or is it really a subset, and can we treat it? I believe that we could at least make some of the symptoms better through addressing GI disturbances, but we still don't know whether it's really part of the disorder itself or simply something that happens more frequently as an accompanying disorder along with the primary disorder.
For the pathogen and host profile studies, what proportion have you got results on so far of that work? And have you found any significant differences yet between controls and patients?
I was just wondering about the oxidative stress and whether you've looked at the damage that can cause, because that can cause new epitopes.
Okay, and the last one, please.
When the human genome was first characterized, one of the sort of repeated stories was that a lot of it was apparently junk. But obviously, since then, the junk story is being gradually eroded by discoveries of functions of various kinds. Do you think that there might be a novel endogenous retrovirus amongst that junk, which we need to be looking at?
Good question. Okay, so I'll do
Watch
three
what you say,
at a
Mady.
time. Okay, tap dancing with, singing at the same time. First question on where are we in the progress of these studies in cases and controls, and I'll focus that we have results now in the peripheral blood studies, not in looking at stool or rectal swabs or nasopharyngeal swabs and so forth. We have completed approximately 80% of the laboratory work side. The bioinformatics for the molecular work is ongoing. It's a complicated story. We have a potential candidate, sort of a novel candidate, maybe, in the spinal fluid, but I can't, we don't know whether that will be confirmed. We have to, pursue that rigorously. And so we are encouraged that there may be some interesting phenomena there, but we don't, it's really too preliminary to state. And the other studies are also just gearing up now. We're going to be working on the rectal swab material, looking at the microbiome, both the virome, the bacteriome, and the mycobiome. And so that's in progress. So the second question was on a look for the degree of oxidative stress and the mechanisms by which oxidative stress may release epitopes. You are right on target. That was sort of the point of the slide that I was showing, is the ways in which the heat shock proteins and the mitochondria and a whole host of host reactions may be perturbed through oxidative stress mechanisms that may be set off by infection, by toxins, even by psychosocial stress, by radiation that we're exposed to, and so forth. So those are very important, and I think that is really key. We are looking at that primarily in the animal model at this time to try to understand how we go from an infection exposure to the proteins of an infectious agent to the antibody result. And so that's also a work in progress, but very key. And the last question is the dark matter question, the dark matter of the DNA. And all of these phenomena that we have, are there potential for endogenous retroviruses? I think absolutely. I think it's but just the potential, we don't know. It's also possible that there are these things called pseudo genes. And these are typically, we call them pseudo genes because they're sort of a product of our own system, and they tend to get randomly inserted. But if they get into randomly, get into a place where it's a promoter is set to allow it to be transcribed and then perhaps translated, one could see phenomena on that part as well. And just by, my simple maths tell me that if you have a particular active metabolic system, you're going to be making more and more of a particular molecule, right? And you have your the possibility that the reverse transcribed, the version of that molecule would be getting inserted all the time. It's just sort of a stochastic sort of thing. How many of these molecules are you potentially making if you have the right enzymes in the wrong place? And perhaps just randomly they could be inserted into a promoter region and actually have activity. That's another possibility. Again,
some people have never believed that there was junk DNA. There's a thing called selection and evolution, and it's all there for a purpose. It's just that we're not smart enough quite often to see the subtleties of the action. So it could be retroviral integration causes a lot of this increase. I've never understood why frogs have more DNA than humans, actually.
Oh, really?
Yeah. I've never understood that. Nobody's ever explained it either, or cared. But anyhow, thank you very much to all the speakers for keeping well up to time.